Now you know that if fertilization doesn't occur, you will not conceive. In such a case, it is possible for you to conceive with the help of assisted reproductive technology using a process called in vitro fertilization or IVF. In this process, the sperm cell and the egg cell are combined in a lab dish for fertilization. After fertilization, the resulting embryo is then transferred to the uterus for implantation. There are many reasons of fertilization not occurring in the natural course. IVF is a successful weapon to deal with this difficulty. In short, it is an effort to duplicate nature's miracle. Over 1 million babies have been born worldwide after IVF. Let's take a tour of the complete IVF process. IVF is done in a stepwise manner. Initially, the ovary is made to grow many follicles at a time with the help of hormonal injections. This is called superovulation, and the follicular growth is monitored by sonography. We hope to get you to grow 5 to 12 mature follicles of the size of 16 to 18 millimeters. At this point, a HCG injection induces ovulation, and the mature eggs can be collected by ultrasound-guided aspiration. This way, follicular fluid containing an egg is sucked out through the needle into a test tube, one by one. This skilled procedure can retrieve 4 to 16 eggs over 10 to 40 minutes. The aspirated follicular fluid is then examined by the embryologist in the IVF lab under stereo zoom microscope for good quality eggs. These are washed in medium, graded for their maturity, and then transferred into the carbon dioxide incubator. After 2 to 6 hours, insemination can be done. The same day, sperms of the husband are separated from seminal plasma by sperm washing. The washed sperms are used to inseminate the eggs. About 100,000 sperms per milliliter are placed with each egg in a separate dish containing culture medium. The dishes are placed in a carbon dioxide incubator at 37 degrees centigrade. 18 hours after insemination, the fertilized eggs are identified. The fertilized embryos left in culture continue to divide and have 2 to 8 cells of equal size with clear cytoplasm after 48 to 72 hours. This is the time when the embryo is ready for transfer to the uterus. It is done on OPD basis with mild sedation. 1 to 3 embryos suspended in a drop of culture medium are drawn into a transfer catheter. The doctor guides the tip of the loaded catheter through the cervix and deposits the embryos into the uterine cavity. This takes 10 to 20 minutes. Bed rest is advised for a few hours after this procedure. By superovulation, it is possible to have many eggs available for fertilization with sperms, resulting in many embryos. These embryos can be preserved by freezing them at -196 degrees centigrade in suspended animation for future use, even for many years.